This one look better? Yes? No? Much better? Good. How many late promoters are there in adenovirus type 2? 1, 2, 3, 5, or 10?
You may or may not have done this.
Two, one, stop. Major late promoter, 1 major late promoter, 5. I should show you this. Just the let's do this again. Somehow these have ended up on the wrong screen again. Today my brain doesn't seem to be working, or at least working just as well as the computer is. So why did some people think 5? It's because the termination, but the promoter is always 1 promoter. So, well, let's actually select the answer here. There we go. So, yes, it's 1 major late promoter. Yes?
Why is there then 3 of the, because there's,
Yes, the tripartite leader. So, that's from what process that you get that tripartite leader? Splicing. So it's a splicing process that you get. So that all gets put together and you get alternative splicing for all of those different late messenger RNAs. And then the 5 different classes are all due to differential what? Termination. So polyadenylation, cleavage, etc. Okay, so now we can get rid of these and actually should score them properly. Good. Okay, try again for the next one. The primer for adenovirus replication is most similar to the primer used for adeno-associated virus replication, RDHV replication, polio, influenza, or Ebola. It's wrong? What's wrong? The answer. Everything is wrong. So, why do people think that it's most similar to polio? Because I said so in lecture last time, yes. What's another reason?
It's a protein primer.
It's protein primed replication. So it's a protein which is used as the primer. What's used as the primer for adeno-associated virus replication? That's why there's not a video for this. What's the primer for adeno-associated virus? What kind of virus is that? It's a satellite virus, but what family of viruses is it? Lots of satellite viruses, lots of different virus families. Helper and satellite, that's, okay, two different things. Yeah, so what family of viruses? These are the parvoviruses. So how do parvoviruses replicate?
It's like herding kittens.
It is like herding kittens, yeah. Replication in parvoviruses is like herding kittens.
They're getting us to answer.
So these guys are really interesting because they are what kind of virus? What flavor of genome do they have? Negative strand. Negative strand what? RNA, no. They're DNA, single-stranded DNA viruses, the adeno-associated viruses. So, how do they deal with replication as a single-stranded DNA? See two different single-stranded DNA viruses on here. What's the difference between those two in terms of their genomes? This is like the last midterm, Dr. Stedman. Why should I care? What's the point? Because these are the kinds of questions that are likely to be on the final, comparison things. So, actually, way back this might have been the first midterm. So, adeno-associated viruses are linear single-stranded DNA viruses, and RDHV is a circular single-stranded DNA virus. So, linear single-stranded DNA viruses replicate with what kind of primers? DNA primers, because they have this bizarre secondary structure at the end, which folds around and gives you 3'OH. RDHV, what's the primer for this? We have absolutely no clue. For poliovirus, the protein. What about influenza? What's the primer for that? There isn't one. The RNA-dependent RNA polymerase can start by itself. These RNA viruses, the RNA-dependent RNA polymerase starts right at the end of the genome. How this starts is a really open question. We don't know exactly how the RNA polymerase can sit down right at the end of the genome and try and figure this out. There was a paper about 3 months ago, literally looking at the polymerase structures for these things to try and get an idea how they sit down at the very end of the genome and start there. It's a really difficult question, and no one really has a good handle on how that actually works. So our answer is yes, polio, protein primed, because Stedman said so in the last lecture. Okay, so today we're going to talk about even bigger DNA viruses, and generally the bigger the DNA virus gets, the more autonomous it is. Basically, it brings more and more of its own machinery with it. And so we'll talk about these herpes viruses this time. Next week we'll talk about pox viruses and miRNAs. These are the really, absolutely ginormous ones. And then also some of the really cool viruses of archaea, which actually have smaller genomes. And then hopefully Dr. Radis will talk about his favorite virus ecology stuff, Hunton viruses later. So yeah, these are bigger herpes viruses, so bigger double-stranded DNA viruses, much larger and, surprise, surprise, more complicated than adenoviruses. But what's really interesting about herpes viruses is that they are pretty similar to bacteriophage, which is another reason that I had that question about all these stuff from way back on previous midterms, because a lot of this stuff is really similar. That's partly why I do spend the time and effort to talk about the bacteriophage at the beginning of the course, because a lot of the replication, particularly the molecular level here, for these herpes viruses is extremely similar to what goes on with bacteriophage replication, and it turns out the structure of some of the capsid proteins are also really pretty similar. So they're probably, in fact, related to each other. A couple of key concepts, as it were, as far as herpes virus replication is concerned. These are mostly the things which are going to be different here relative to the other viruses that we've talked about. Herpes viruses have this fascinating latent infection process. And again, this is mostly what they're known for, cold sores, etc. These are replication of virus at kind of random times well after infection. So this latent infection process, it's reasonably well known how this works, but turns out it might actually be really important for developing vaccines and so on and so forth that we'll talk about right at the end. Herpes viruses are really different, in fact, than any other kind of virus, is that they're enveloped viruses, but there's a specific component between the envelope and the capsid, so not just like a matrix protein, most of the stuff we've talked about so far, that has some very specific proteins. It's called the tegument. And so it's sort of this region between the envelope and the capsid, which has some very specific proteins, which, as we'll see, are really important for virus replication, particularly this VP16 protein, which is present in the tegument. Really strong transcriptional activator. The genome is unlike any of the DNA viruses we've talked about so far in terms of how it's arranged. It actually has this really strange recombination that seems to take place in the genome. And then there's, in big contrast to the adenoviruses, very, very little splicing that takes place in these genomes, as opposed to adenovirus, which is, of course, splicing all over the place. So again, we'll talk about where it comes from, a little bit about the structure and binding and entry, but mostly about transcription and replication as far as this is concerned. So where do they come from? Most of the studies that have been done on herpes viruses have been done on HSV1, also known as herpes simplex virus 1, and so most of the rest of this talk today, my lecture is going to be on what's been learned about HSV1 in terms of the genome and how it replicates. The molecular virology here has best been done on HSV1. Very closely related are what most people think about when they think about herpes. This is HSV2, very closely related to HSV1. Chickenpox is another one of these herpes viruses, or say it's caused by one of these. Mono, Kaposi sarcoma virus, Cytomegalovirus, et cetera, et cetera, et cetera. And one of my colleagues is now just down the road at Oregon State, works on coral viruses, and there are quite a few herpes viruses that are infecting corals. So this all ended up on, CNN and I think some of the other less reputable maybe news sources. Look, the corals have herpes as well. So I didn't bring out any of the cartoons that were done at that point, but it's interesting. But again, one of the really interesting aspects about these herpes viruses is this latent infection process, where they basically go into the host and hang out, and potentially for the life of that particular organism. Once they're there, they're there. It's not like the case with retroviruses where they have to, but they just seem to basically hang out in the nucleus, get replicated, and stay at a very low level. And then the induction of production of these is a really interesting question, and one of the things that people really don't understand, but is a very active area of research. And one of the things I also wanted to point out on here is this cytomegalovirus, which I mentioned briefly before, cause of mononucleosis, but much more importantly, somewhere between 50 and 90% of people are infected by this virus with almost no symptoms whatsoever. So it also does some really cool things as far as the immune system is concerned, which, as I usually say, is way beyond my pay grade, but our seminar speaker in biology on the 5th of June is going to talk about this. Really, really cool stuff. Cytomegalovirus completely tweaks our immune system in some really fascinating ways. But we're mostly going to talk about herpes simplex virus, again, because it's a much better understood one, mostly because it grows a lot faster. It's a lot easier to work with than some of these other ones, which take ages and ages and ages to work with. So that's just like we've talked about in many other cases. The reason you work on particular viruses is because they're easy to work with and hopefully are model systems for everything else. So, yeah.
Do those lesions come back in the same location every time when it's latent and it gets pre-initiated?
Yeah, so we'll talk a little bit about, so the question is, do the lesions always come back in the same place? We'll talk a little bit about that. It's not entirely clear what that is, but it's usually as true for all viruses. You're infecting one particular cell, and so that one cell is then going to have to reproduce. But then those viruses can move from cell to cell. So it's going to depend a little bit on where that is. So what's in the virions that we'll talk about in just a second. One of the interesting things about the HSV1 genome, and this varies now from herpesvirus to herpesvirus, is that it's this whole mixture of 4 different genomes. Each virion has one of these, again, incredibly creatively named long and short segments, where these 2 segments can be either arranged in a head-to-tail fashion, a head-to-head fashion, a tail-to-tail fashion, or completely flipped around in the other orientation. And that's because of these repeated sequences, like inverted repeated sequences, which are present in the HSV1 genome. And so these repeated sequences seem to be important for this flipping, mixing, and matching of all of these, probably important for recombination and particularly important for packaging. When we look at packaging, which again turns out to be extremely similar to what happens in a lot of bacteriophage, that's due to these repeated sequences and how they get put together here. And the nomenclature here is A for your repeated sequence, and then B for a repeated sequence is in the large subunit, C, which is in the small subunit of the genome, and then prime just represents the complementary copy of that. So again, flipped over a lot like the ITRs that you have at either end of the genome and adenovirus, only you've got to put 2 of them together here, and they can be flipped around and mixed and matched. In between these repeated sequences, you have, again, ridiculously creatively named unique sequences. So you've got unique sequences in the large segment and unique sequences in the small segment. This is a really big genome as far as viruses are concerned. In fact, it's the largest one that we've looked at so far, about 150 kb. And just to prove that it's really big, here's a micrograph where we've got the FIX174 genome right here, this little black line. Again, single-stranded circular DNA virus. And then here's our herpesvirus genome, starting over here and going all the way over here and winding around over here and getting back over here. And you can literally measure, FIX174 is about 5,000 bases in length. So this is 150,000 bases in length. You can literally measure along here and you end up with that number. Yeah?
On that last slide,
Yeah.
does that particular layout of A, B, B prime, A prime, C prime coincide with one of those 4 patterns you've got below it, or is that just always how they're doing?
Okay, so the question is, this particular cartoon here, is this the way that it's always laid out? So this is the organization up at the top here of the first line. And so if you have the second one, here you'd have A prime, C, C prime, A. That's the way it's flipped around. And so that's what's meant here by that, the arrows at the bottom. Of course, you can't tell that from here because you don't know what the sequences are. But you just know that it's a really long genome, which is present here. What's coded for in this genome? Looks ridiculously complicated. It is. But I've tried to highlight the important genes, i.e. the ones we're going to be talking about here, in more detail. So this is drawn as a circular map as opposed to a linear map, and that's because these are repeated sequences, and so we don't really know exactly where that's going to be. And it turns out when these guys replicate, they also are going to replicate as circles or rolling circle replication. So here we've got our unique small sequence, all the unique large sequences, and then all the names of the open reading frames, unique large 46, 43.5, 226, 26S, just a god-awful mess in terms of all of these different sequences. Same thing here, all the unique sequences, U, S, and all of these unique ones. Much more interesting are, of course, the ones that I've circled, which are present in some of these repeated sequences. So here, this is alpha 0 that we'll take a look at, alpha 4. It's in a repeated sequence, so you're going to have alpha 4 here and alpha 4 over here. Two copies of this gene. Same thing is true for alpha 0, alpha 47. These are going to be all things which are present in these particular repeated sequences. We'll also talk about VP16, which is here in one of the large segments, and this VHS protein. Everything else you can basically ignore except for the blue arrows. The blue arrows represent the lat for latency transcripts, which we'll talk about really toward the end. So I decided to switch around, talk about the genome first, and then talk about the capsid, because it's important where some of these proteins actually are in the capsid. So here's the cartoon of a typical HSV1 capsid. Here's the electron micrograph that corresponds to it down here at the bottom. So you've got your genome on the inside, again, about 150 kilobases, kilobase pairs, I should say, in size. This is a pretty big piece. Packaged inside a very regular icosahedral capsid made up of VP5, and you can do the math here, figure out what that triangulation number is. How would you do that? Divide by 60, exactly. And about 5 other proteins that are present just in the capsid. The outside, again, this is an envelope virus. We've got a bunch of glycoproteins on the outside. There are a huge number of glycoproteins relative to all of the other viruses we've talked about so far, present here on the outside of these virions. That probably has to do mostly with interactions with the immune system. And again, this is something that Dr. Frew will talk about a lot more on June 6th. But it's a very large number of glycoproteins that are present here on the outside. And then the unique thing as far as all these herpes viruses is concerned, it's what's in between. So it's what's in between this envelope and the capsid. We've got at least 14 specific virus proteins that are in there, messenger RNAs, etc. And so what that means is as usual with an enveloped virus, what happens? The envelopes fuse, wherever it happens to be. It's slightly different for different herpes viruses. But once you have membrane fusion, then you have release of all the proteins which are in here into whatever compartment you've fused into. And so this is not unlike what happens with things like the retroviruses, where they're bringing in all of these extra enzymes with them as well. Only here the enzymes are not in the capsid, they're on the outside, they're in the tegument here. You can look at this in a little bit more detail. This is a cryo-electron image reconstruction here. Here's a 5-fold axis. Shall I break the pen back out? Probably should. So, anybody done their 960 divided by 60 yet? Okay, so what does that mean? What should we have? Oops, come on. It's 10. So, how do you get your number to be 16? 4 from one 5-fold axis of symmetry right here to the next 5-fold axis of symmetry right here. How do we get there? We go 1, 2, 3, 4. So nice, regular, icosahedrally symmetric particle, again, mostly from that VP5 protein, which is there, and certainly the way the capsid is put together, it's that complete quasi-equivalent structure. There is one slight exception to this, and we'll see when we talk about packaging. There's a portal protein, so one of the 5-fold axis of symmetry is in fact a little bit different. How do we get in? Proteoglycans on the outside of the cell. These guys fuse at the plasma membrane. The capsid is released. It gets transported to the nucleus together with some of these tegument proteins. Again, the tegument proteins are those that are between the envelope and the capsid. And once it gets to the nuclear membrane, the DNA is released inside the nucleus. It seems the capsid actually stays on the outside, and we'll see that in just a second in some micrographs. So again, fuses at the plasma membrane, transported here. There's some wonderful animations. I didn't pull them up with Dianin sort of walking along the microtubules, dragging the virion with it here. So that's how you get in. Once the genome is released, actually not at all dissimilar to what happens with the bacteriophage. It seems to be released through that portal again, but now into the nucleus as opposed to the bacteria where you're injecting it across the membrane on the outside of the cell, becomes circularized and just seems to hang out in the nucleus. So what does this look like in a TEM process? Here we have a herpes virus. Here's the capsid on the inside. There's the envelope. Here's the tegument in between. Fuses, hang on, this is going this way. Here, outer membrane, fused with the plasma membrane. You have release of the capsid. That capsid gets inside, then slowly starts to get transported through to the nucleus, where you see these empty capsids and all of the genome and the genome proteins which have now gotten into the nucleus. So fusion out here at the plasma membrane. You can also see that this capsid still has DNA in it or something in it, whereas it's now empty once you finally get to the nuclear envelope and in there. The genome, once it gets inside the nucleus, is, of course, transcribed by cellular RNA polymerase II. You've got 3 sets of genes here, and just to make it more confusing than what we've always talked about before, early, middle, and late, the herpes virologist said, oh, we'll call it immediate early and late. Again, I would just call this early, middle, and late. They also, because too many people work on these herpes viruses, we'll call them alpha, beta, and gamma. And that's very often what you'll see in terms of some of the proteins. So the immediate early proteins is what gets expressed most early. Beta is the next ones, is usually what you think about in terms of our early proteins. So normally we've talked about the early proteins mess around with the host, then you make more of the genome, then you make, say, the viral genome, and then you make more of the virion proteins. And so that's basically what happens here. Mess around with the host, make your genome, and then put everything together here. So almost all of these transcripts, interestingly, are unspliced. Now, why they're unspliced is, again, kind of one of these why questions. You should never ask why questions in biology. But it seems that that splicing process is something which takes, of course, quite a long time in cellular mRNA processing. The virus is going to be able to make many more of its own RNAs relative to the host RNAs if it doesn't have to bother with the whole splicing process. Some exceptions here are these very early transcripts which are made. This, again, terminology here is horrible for herpes viruses. ICP just stands for infected cell protein. And the first ones, third ones, et cetera, what you find. But what you notice is that in these repeated sequences, both at the end of the large segment and at both ends of the small segment, this is where you have a lot of these immediate early proteins which are made, but also where the latent transcripts are made. And so these are then those conserved things. So what you have, immediate early, and late, not surprisingly, this is where you see the transcripts. They're also being regulated. How do they get regulated? Basically through the activity of various different viral proteins and lots of cellular regulators. So this is what a typical regulatory sequence looks like in front of one of the immediate early promoters, and this is in particular the immediate early promoter for ICP4. What do you see in here? Lots of different binding sites, SP1, TATA box, etc. which are cellular transcriptional regulators, but you also see all these binding sites for this protein called alpha-TIF and also ICP4. ICP4, again, infected cell protein, so this is a viral protein which is going to bind there, and alpha-TIF. What the heck is alpha-TIF? I know it as VP16. Again, unfortunate, all the nomenclature here. VP16 and alpha-TIF are exactly the same protein, present in the tegument. And what does that mean about that particular protein, that particular transcriptional activator? It's there immediately. You don't need to transcribe it. You don't need to translate it. That protein is already there. So that transcriptional regulator is already available and ready to transcribe all of these genes that need to be transcribed right at the beginning. This particular one is an activator together with two cellular transcriptional activators, OCT1 and HCF1, and we'll take a look at those a little bit later on. So, this is true for these alpha or immediate early kinds of genes. Your early genes are mostly regulated by cellular and, to some extent, some of the viral proteins as well. Your lates are all pretty much just cellular proteins which are regulating that. And what that tells you is that all of the cellular messenger RNAs are basically no longer functional, even as soon as you get to your early transcription phase. How does that work? Well, you have this VHS protein, which leads to RNA degradation. It's also present in the tegument. So basically, there's this race between making the viral messenger RNAs and the cellular messenger RNAs. The viral messenger RNAs are getting made faster and faster. They get made quickly because they don't have splicing that has to take place. And therefore, your degradation is going to happen more for those RNAs which are sitting around longer before they go out into the cytoplasm and get translated. VHS, of course, degrading all of the RNA would be a bad idea. It ends up getting blocked by new production of VP16. So this is a case you block hosts by cutting down the RNA. Once you have ever completely taken over that host, the only thing it's doing is making more virus, then you don't want to degrade the mRNA because it's all viral mRNA. And so once you've made new VP16, that will stop that process. So what is this VP16 protein? This is, in fact, the sort of the classic transcriptional enhancer protein. The VP16 protein, in effect, was one of the very first models that people used for studying transcriptional activation because it was this really, really good transcriptional activator. Has a acidic activation domain, so split up, different domains in your different regulators here. It works extremely well when you hook it up to a separate DNA binding domain. Stimulates basically everything that you need to do in terms of transcription, put together the whole transcription complex, it stimulates elongation, but probably most importantly, it gets rid of nucleosomes. So what does that look like in a cartoon fashion? Here we have our VP16. This is a completely artificial construct where it's now attached to a separate DNA binding domain. So this VP16 can now bind to DNA. It binds to DNA, stimulates formation of the pre-initiation complex together with TF2B, and also moves nucleosomes around in such a way that you can get this pre-initiation complex. This is how people showed how this was in fact working, what this particular activation domain on VP16 was doing, but that was all well and good except for the fact that VP16 by itself doesn't bind to DNA. So how is it actually working in a viral infection process? Well, the way that works is that it needs cellular proteins to bind to DNA, and these are cellular proteins which are usually transcriptional regulators in and of themselves. So this is the OCT1 protein, has a homeodomain, which is the eukaryotic equivalent of the bacterial helix-turn helix DNA binding motif. So homeodomains bind to DNA. these guys interact through protein-protein interactions with VP16 and another cellular transcriptional regulator. And this right here is a really gangbusters transcriptional activator. Again, so it's the VP16 together with these cellular DNA binding proteins or transcriptional activators, which lead to activation of all of those alpha proteins. What are the alpha proteins? These are the first ones which are getting made. This is not stuff which is in the tegument. These are the genes which are getting transcribed. Infected cell protein 0, infected cell protein 4, number of these. But the main thing that these guys are doing is activating transcription, activating gene expression, enhance expression of late genes. Here, blocking RNA splicing. Why do you want to block RNA splicing? Because your genes don't have splicing in them. And this one is important for regulating the immune system. But here, the two that we really care about, which is why I write them over here, and I would think about remembering them for exams. ICP4 is a regulatory protein. Also, if we back up, I don't know, backing up here is such a good idea, but we go too far. You'll notice that the ICP4 promoter also has ICP4 binding sites, particularly this ICP4 binding site, which is right on top of the transcriptional start site. So once you make a whole bunch of this protein, it shuts off its own transcription. So it's an autoregulator, or in this case, autorepressor. And then ICP27, again, blocking splicing. So these are just some of the alpha proteins, and I've reduced them from the table down to these couple, which I think are particularly important. So alpha proteins in particular, ICP4 is an autorepressor, but it's also a gene activator together with ICP0. You make these as your immediate early proteins. What do you think they're going to regulate? They're going to regulate the early proteins. What are early proteins important for? Genome replication. So genome replication in herpes simplex virus have origin binding complexes, single-stranded DNA binding, helicase primase, DNA polymerase, processivity factor. Basically, everything you need to get replication is encoded for in the virus genome. There's very, very little that these viruses need from the cell in terms of getting DNA replication. The RNA stuff, again, that was all cellular RNA polymerase II, cellular transcription factors. DNA replication, a lot less so. So how does it work? Turns out it's actually extremely similar to pretty much any kind of DNA replication that we've talked about. We have origin binding proteins. There are 3 of them in the virus genome. Doesn't seem to matter whether you have 3, why you have 3. Just look at one of them here, with this origin binding protein, helicase activity, primase activity, DNA polymerase activity, and sliding clamp activity. All of them which are viral proteins. I'm not saying the names of them because I can't remember them. If I can't remember them, I can't expect you to remember them. Can I? No. It's been a long week, I guess, for everybody. So it doesn't really matter which of these origins it is. You start out doing this theta replication, which is going to start out at one origin, have bidirectional replication, goes around the genome. You have to resolve the concatenamers once you get to the end. That's what happens in early replication. Later on in replication, this shifts over to doing rolling circle replication. Rolling circle replication is where you have your double-stranded piece here, and hopefully George talked about this a little bit with RDHV. You have a nick in one of these strands. That then serves as the primer for the cellular polymerase. It just goes around and around and around and generates a single strand. In this case, because these viruses encode all of these good primases, etc., you'll then make a copy of that second strand, so you end up with a double-stranded piece, which is multiple copies of your genome, all attached end to end to end with each other. probably that's where the recombination takes place, at least in HSV1, with these repeated sequences and inverted repeated sequences which are there. Another thing which I wanted to remind myself of is it's not just the enzymes here which are important for replication of the viral genome, the primase, the polymerase, etc., but also this viral genome encodes thymidine kinases, ribonucleotide reductases, and uracil N glycosylases. What do you need these proteins for? What's thymidine kinase do? Phosphorylates thymidine. Why do you need thymidine? DNA replication. Most cells are not undergoing replication most of the time. Do they have a whole bunch of deoxyribonucleotide precursors around? No. But if you want to make sure that your virus genome gets replicated, you want to get these ribonucleotide, just RNA pieces, convert them into DNA, and make them into thymidine. So you have a ribonucleotide reductase. What do ribonucleotide reductases do? They take off the 2' OH, so you go from RNA to DNA. Thymidine kinase. And then, what's really, to me, is pretty amazing, they also encode, this is a viral uracil N glycosylase. What do uracil glycosylases do? DNA repair. So they take out uracils that get put into DNA by mistake. And so this virus genome actually encodes a DNA repair enzyme, which is really pretty amazing that it does that. So we've replicated our genome. Now we need to make our virion. These are now our late genes. So here, late genes. To express late genes, you have alpha proteins, which are regulating these. And to really make these late, they also talk about in the book a little bit about these early late genes. The real late ones, you have to have DNA replication before these genes are actually transcribed. Once you've made all of these genes, and actually I don't have, I forget which one's VP5, I think UL38 here might be VP5, but not critical. You make a lot of these proteins, but it's not just that one protein that you need to make the virus structure. Turns out that these big, what was it, T equals 16 icosahedral capsid shells, putting them together requires a lot of extra proteins, and these are scaffolding proteins. And the scaffolding proteins are extremely similar to what you have in a lot of the bacteriophage, which are putting together these large icosahedral structures that they're then later going to be packaging their DNA into. So a lot of these late proteins get put together and basically make a scaffold or a template around which the whole capsid is made. So that's diagrammed here. Here are these proteins all coming together. You end up with a procapsid. Again, very similar to what you see with bacteriophage. Then the scaffolding protein gets degraded. It degrades itself very conveniently. And now we have an empty capsid that the DNA gets packaged into. This is, of course, now the capsid. You also are going to need to have the envelope put around the outside. But first, let's look at how the DNA gets in. The DNA gets in from one of these concatemers, which has been made here through rolling circle. So rolling circle, the polymerase is just going around and around and around here, making this really long, multiple copies of your genome here. The second strand is made by the viral polymerase, again with its sliding clamp-like factor. Here we have our empty capsid with now the portal at one end. So again, it's not completely symmetric. This portal protein even has sequence similarity to some of the portal proteins that you find in bacteriophage. One whole genome gets in here, basically from one repeated sequence to the next repeated sequence. Once you've filled up the capsid, there's cleavage again of the DNA, which looks really similar to what happens with all the bacteriophage. And you have a now filled capsid. That filled capsid has to get, oh, and the protein that does that is a protein very similar to the terminase that we talked about when we talked about bacteriophage way back when in the depth of time. So now we have our capsid, which has been assembled in the nucleus. Now it needs to get out. Most people seem to think in the book, they say there are these 3 different models about how it gets out. Most people seem to think that it's this model 2. And the way this works is that the capsid, instead of going out through a nuclear pore, which is way too small to get one of these T equals 16, ginormous things, got 150 kilobase pairs of DNA inside it, to get out, it seems to butt into the nuclear membrane space right here and pick up some of the tegument proteins which are there, but strangely enough then fuse on the other side of the nuclear membrane, then go through the Golgi, butt into the Golgi, and now you've actually got 2 membranes. So this is the vesicle membrane out here. Here's the membrane of the envelope of the virion. This then vesicle will fuse out here at the plasma membrane, and then you have a released virion, which is outside there. So you've basically gone through multiple membranous components in order to finally get out here and be released. This is the one which seems most bizarre because you bud here, then you bud out, you bud in, then you bud out again. But most of the experimental evidence actually lead towards how this is in fact happening, how you're putting these virions together. So this is your classic normal replication of herpes virus. Binds, gets inside the cell, gets transported to the nucleus, you have your immediate early, your early, and your late genes, everything gets out. But what's one of the really fascinating things about these herpes viruses? They go through latency. So what's happening with latency? Latency, at least for HSV1, is almost always in neurons. So what happens is you have this infection process, you fuse, the capsid is released, that capsid then gets transported to the nucleus. For neurons, as you guys probably know better than me, and I don't know if you talked about neurons in the cell biology exam, they can be ginormous. Some of these giant squid things are like meters in length. And so what happens is the virion fuses the membrane, you have the capsid, that capsid walks its way all the way along the microtubules, all the way down the axon, and ends up at the nucleus. And so this is some of the, unfortunately I don't have any of these pictures, but you can label the capsid and actually watch it being moved along the axons inside some of these neurons. It's really pretty amazing. So in that process, you seem to lose some of the tegument proteins. Again, it's not terribly surprising. They're sticking on the outside of the capsid. And so as you're moving through the whole cell, that seems to be lost. Now remember, VP16 is this really critical transcriptional activator which you need for a lot of the alpha, the immediate early genes which are being expressed. So you don't have those anymore. You're not going to be expressing all the proteins to make more of the virus. So then you start making these latency transcripts, the ones that were labeled as LAT before, and these are pretty stable transcripts. They don't seem to encode any protein, but they're absolutely critical for latency. You knock out these transcripts, then you don't get latency at all. They, unlike, again, most of the normal viral messenger RNAs, don't have introns, except for one which is stably spliced out. And you can actually see the intermediate, the splicing intermediate. So splicing, you have a 2' hydroxyl in the middle of your intron that the end of the intron loops around, forms a covalent bond with, and then that gets released. You can actually see these circular or lariat kind of structures in infected cells here. What this is doing, again, you'll notice lots and lots of question marks here. Are they antisense? Are they important for protecting against apoptosis? How do you do induction? It's a very, very open question which is going on here. A lot of people have been looking at what happens with small RNAs. In fact, literally in the last 10 years, people have finally realized that small RNAs are important for regulation. Turns out that there are a number of small RNAs encoded in the Herpesvirus simplex 1 genome, which are important for regulation of not just cellular proteins, but also a number of viral proteins as well. Yeah?
So that's one slide.
Yep.
So with the replicate transport, does it impose some kind of sorting signal then that tells us also where it goes?
Yeah, so the question is basically, is there some kind of sorting signal that says capsid should go to nucleus? And that's exactly what it does. I didn't mention that when we talked about the original entry. So in normal cells, the epithelial cells, which these are normally replicating rather than in a neuron, those have a specific nuclear localization signal on the capsid. It's not on VP5, it's one of the other proteins which is there, and that says, okay, transport this to the nucleus. So it has a specific nuclear localization signal on it, part of the capsid. Okay, so these small RNAs as well. There are a number of animations here. We could either go over them briefly. I think they're a really nice review of pretty much all the things we've talked about in terms of the replication, transcription, etc. Most of the proteins that I've emphasized here are also nicely emphasized in this particular case. Do you want to go over them now, or do you guys want to go over them when you're studying for the exam and it's Friday and we're falling asleep already? We'll just have a quick vote. Now, later. Now, later. Okay, we'll do some of them now. Just a couple of things to look at here, and this is just a nice way of looking at how a lot of these things are functioning here. So this is the homepage of the unfortunately late Ed Wagner down at UC Irvine has been studying herpesvirus for years and years and years. What I think is really cool is the university's kept his website up, which is really nice here. So HSV structure, receptor binding, transcription, latent infections, DNA replication, encapsulation, and release. I'd like to talk about latent infections here because I think I did a pretty crummy job of doing this before. Here's our latent infection. Here's your neuron. They bind, and in fact, there literally are movies where you can see these capsids being transported to the nucleus. So here you have transcription of these latent transcripts, which happens once you have the genome that gets in. Those latent transcripts then splice out these stable introns, which again can be found inside the cell. You also have the chromosomes now. Here, this is the viral genome, which is associated with individual cellular histones. So that's what's happening in latency. If you look at the HSV structure, here, this is a cryo-EM of that structure. Here's the cartoon, genome in the middle. Here's our capsid. Here are all of our tegument proteins here on the outside, envelope proteins, how they're associating with the envelope. How does this get inside the cell? Here, this actually should have the envelope around the outside, receptor binding, coming inside the cell. Here we have the glycoproteins on the outside interacting with cellular glycoproteins. Magic happens and you have membrane fusion, and the capsid is released. This then gets transported along the microtubules to the nucleus. The viral DNA is released, and he calls this alpha TIF instead of VP16, but it's that really critical transcriptional regulator which you have here. Once that happens, you go inside the nucleus. Not sure about all the animations here. Immediate early, followed by late. With the numbers here. Here we have binding by alpha TIF, which again is VP16, together with the cellular transcription factors. And then you transcribe all of these immediate early genes. Those early genes, particularly the alpha 4, which is this transcriptional regulator, leads to production of the early genes. Those early genes are the ones which are important for genome replication. Genome replication, these are all of your proteins that are coming back in, leading to genome replication. Once you've done all this genome replication, now you have these final late proteins. These are all the ones which are going to be making your capsid. Those get re-imported back into the nucleus. You assemble each of the capsids. Replication happens through DNA separation here. Here's your HSV genome. One of 3 origins gets used. Again, you can use any of the 3 origins. It doesn't seem to make a difference, again, because this is a circular genome. Your origin binding protein functions, again, very similar to origin recognition complexes that you have in eukaryotic cells. Then you have all of the viral proteins which come in and replicate here. Again, it's an RNA primer made by a virally encoded primase, DNA polymerase, together with a processivity factor. Again, all of these are viral proteins. It's actually described down here at the bottom in text exactly what's going on here as well. Then you have this normal theta replication followed by nicking, and you have the polymerase which is going to extend here. They're your little nucleocapsids that get packaged. These 2 little dots here that are coming in, that's the terminase, which is chopping up the DNA and putting it into the capsid. A little bit more detail on the encapsidation process here. Here are all of our capsids. We need to get genomes inside each of those. We enter it. Here we have our nucleus, which has all of our genomes in it. Here are our virus-encoded proteins. Most of these are scaffolding proteins. So again, you make this scaffold to start with, and then you put VP5 on the outside, which is mostly what's making this whole capsid together. These then get degraded, and then you package your genome inside of those. How does it get outside of the cell? And this is again the model 2 from our textbook. You have the capsid, which has been made, coming out of the nucleus. It seems to go into the nuclear membrane. You have the viral proteins here, about 10 of them again, which are in the envelope. Those then, here's our capsid coming out here, associated with all the tegument proteins. Again, VP16, not alpha-TIF. If I do have something like that in an exam, I'll try and have both. That then buds into these exocytotic vesicles. So now we've got 2 membranes. We've got the envelope of the virus here, the vesicle on the outside there. This now goes to the membrane and releases, because you fuse this membrane, that's the exocytotic vesicle membrane. These guys go off and try and find some new cells to infect. So there's one more thing in the PowerPoint that I wanted to mention, which I sort of talked about right here at the beginning, and that has to do with Cytomegalovirus. Some of you may have heard about Louis Picker. He just got, I think, $25 million from the Gates Foundation for HIV vaccines, some very, very promising HIV vaccines that work really well in simians, which is why he's associated with the Oregon National Primate Research Center here, and also the Vaccine and Gene Therapy Institute. Klaus Fru, who's also at the VGTI through HSU, is president of Cytomegalovirus, which is a company which is trying to use this cytomegalovirus backbone for developing new vaccines. Just got an email from the president of the company, actually the CEO of the company, he's the president, and he'll be giving the biology seminar on the 5th of June, so the day before the last day of classes here. So great review in terms of these cytomegaloviruses. I'm not going to put stuff from his talk on the exam, don't worry. So should be a lot of fun. So with that, those are your herpes viruses. Have a great long weekend. There will not be lecture on Monday. You're welcome to come. There won't be clicker questions. I won't be here.